Today we're going to be talking about how cells are driven into apoptosis when they are exposed to a toxic chemical that triggers a specific type of DNA damage. And the chemical that I will be talking about today is Etoposide. I have mentioned this chemical in a previous video. It's the kind of chemical that triggers double-stranded DNA breaks in the DNA. Double-stranded DNA breaks. And this is due to the fact that it actually inactivates topoisomerase 2. And topoisomerase 2 is one of those enzymes involved in DNA replication that helps untangle DNA as the replication fork is moving along the template. So the activity of Topo2 is essential. It actually does double-strand breaks in the DNA, but then it has an enzymatic activity that allows the religation of the DNA after it has done the untangling. So it breaks and then it religates the DNA. But if the second enzymatic activity, the ligase activity, is inhibited, then it won't be able to fix the DNA that has been cleaved, therefore producing double-strand DNA breaks that remain in the genome. And as you can remember, this is the kind of things that are classically associated with DNA damage. It's not good for the cell to have double-stranded DNA breaks. So if you treat cells with this chemical at the right concentration for the right amount of time, you're going to generate this kind of damage, double-stranded DNA breaks. So how could this actually trigger cells to go into apoptosis? So first of all, we need to be aware of one thing, and that is at what particular stage of the cell cycle is this agent going to produce the kind of signal that will hold progression through a cell cycle. And it is well characterized that this kind of damage is going to produce a halt in the progression between G2 to the M stage of the cell cycle. So G2 to M will be halted. How could that be? Well, to go from G2 to M, the kinase that is essential at that level is the CDK1. And CDK1 has a cycling partner, just like all the kinases drive progression through a cell cycle, and that partner is cyclin B. So you have CDK1, cyclin B. Cyclin B is produced only during the G2 stage of the cell cycle. And when cyclin B is produced, it associates very rapidly with CDK1, and this complex now becomes very rapidly phosphorylated. This phosphorylation is required for the activation of the complex. That phosphorylation is made by the CDK activating kinase, CAC. It phosphorylates CDK1, and that activates the complex. But the moment that this phosphorylation event takes place, there is another kinase that also acts on the same complex, a kinase known as WEE. And in this case, WEE is going to produce the kind of phosphorylation event that will be inhibitory. So let's make the distinction between activation and inhibition using that other symbol. So WEE is going to also phosphorylate CDK1. This is going to phosphorylate CDK1 in two additional places. And that is inhibitory. So to indicate that this is an inhibitory phosphorylation event, let's put it in another color. Let's use pink for that. All right. So in order to allow for this complex to be fully active so that it can actually drive the cell into mitosis, this requires then the activity of a phosphatase. And that phosphatase that will eliminate these inhibitory phosphorylation events is CDC20C. So that phosphatase needs to be active so that it can actually eliminate those phosphate groups. But this phosphatase becomes inactivated when there is double-stranded DNA damage. Why? Because there is another kinase that senses that kind of damage. You have the double strand, you have the break in the double-stranded DNA molecule, and that activates the ATM kinase. And ATM kinase, when it is active, it phosphorylates the check kinases. There are two different types of check kinases, and these kinases are able to then phosphorylate this phosphatase. So they do act on this phosphatase, it becomes phosphorylated, And this phosphorylation event is going to inhibit the activity of this phosphatase. Now, this is what is going to keep the cells from being able to progress into the M phase, because this kinase now remains in the phosphorylated stage, and this phosphorylation is inhibitory, therefore is the kinase complex as a whole is inactive. Another side of the activity of the check kinases is to phosphorylate another target. And the other target that is phosphorylated by the check kinases is a protein known as p53. So as long as the double-stranded DNA damage remains in place, the check kinases will be phosphorylated by ATM, and as it is phosphorylated, it'll remain active. So these check kinases are constantly remaining active, which means they are phosphorylating not only the CEC20, therefore halting progression through the cell cycle, but they are also phosphorylating p53. And this is a very important target in the whole process because p53 will normally be degraded. So it normally will be a target for a ubiquitin ligase that will polyubiquitinate p53. So you will have a polyubiquitination event here. And that polyubiquitination event typically results in p53 being degraded by the proteasome. Degradation by the proteasome. However, when p53 is phosphorylated by the checks, this phosphorylation event that is mediated by the check kinases now is going to protect p53 from being polyubiquitinated. So this event will not take place, there will be no polyubiquitination, and therefore p53 will be stabilized. And once p53 becomes stabilized, it becomes an active transcription factor that will go on into the nucleus and activate the transcription of different targets because it has affinity for different promoters. So some of the promoters that will be targeted by p53 include the promoter for p21, which is an inhibitor of the CDK2 and cyclin E complex. This is required to enter into S phase. So it's not really relevant here because the cell is stuck between G2 and M. Remember, that's where the cell has been halted in its progression through the cell cycle. So we have p53 hitting this target first, but if there is further accumulation of p53, then p53 will start hitting other targets, other genes. So it'll be actually binding to a promoter of other genes. And the one that will be relevant now is the promoter for a gene known as PUMA. And PUMA is one of those pro-apoptotic proteins that when it becomes activated, now it will be then produced in substantially higher levels as it may normally be found in the cell. So PUMA is now being transcribed and the protein is being produced. And now when PUMA is produced in enough quantities, it'll start then interacting with BCL2. And BCL2 is a protein that normally has the ability to interact and inhibit one of those BCL2 family members that are pro-apoptotic, and that is the BAX protein. So BAX is one of those proteins that when it is on its own, it has the ability to form a protein complex that forms pores. So it's a pore-forming protein, BAX is. But it won't be able to form pores for as long as it is associated to BCL2. But now, since the cell is halted in its progression through a cell cycle, p53 has accumulated to high levels, it has now promoted the transcription of the PUMA gene, and now PUMA is being synthesized, the protein is being produced, This protein now will compete on its binding with BCL2, so it will interact with BCL2. Therefore, Bax will be released. Bax will now be free from its inhibitor, and it will now be able to form pores. Those pores will be formed where? Well, they will be formed on the mitochondrial membrane. So now you have pores in the mitochondria, and inside the mitochondria you have cytochrome C. So now cytochrome C will be able to flow out of the mitochondria into the cytoplasm, and that flow of cytochrome C out of the mitochondria and into the cytoplasm will now allow the formation of complexes with the APAF protein, which will then lead to the formation of the apoptosome. The apoptosome will now become active in the presence of cytochrome C, and it will then lead to the cleavage of the first caspase, caspase 9. Once caspase 9 is activated, it will then activate caspase 3, and this will trigger the cell fully into apoptosis, because once caspase 3 is fully active, it will go on and start cleaving a different array of cellular targets that will lead the cell into apoptosis. Now, besides cytochrome C, there are also other proteins that will be leaking out of the mitochondria, and the most important of them is a protein known as Diablo. Interesting name for those of you who are Spanish speakers. Diablo is one of those inhibitors of apoptosis. So there are proteins in the cell known as inhibitors of apoptosis, or IAP proteins. These proteins prevent the full activation of caspases. So they have the ability to inhibit caspases, and as long as they are active, they will neutralize the effect of this cascade of events that has been indicated here. But now, this inhibitor of apoptosis will be neutralized in its activity because of the presence of Diablo now. Diablo is an inhibitor of the inhibitors of apoptosis, so therefore now the cell is free to go fully into apoptosis. Okay, this has been a relatively short video, but the main idea here is to be able to link events that trigger specific types of DNA damage, exposure to Etoposide, which is again an inhibitor of topoisomerase 2, which results in the accumulation of double-stranded DNA breaks within the cell. So what I want you to do is link that event, the accumulation of double-strand breaks in the cell, with two main events. One of them is halting progression through the cell cycle at the G2M interface, and the second event is triggering the accumulation of specific factors within the cell that lead to the activation of apoptosis. So that's the main idea that I hope you'll be able to take out of this video, and I hope that this diagram does not turn out to be too confusing for you guys. As always, if you like this video, give it a thumbs up, and remember to subscribe to my YouTube channel.